Hello, and welcome back to Beyond Networks, the evolution of living systems. We're right in the middle of our module on dynamical systems theory and how we can use dynamical systems theory to characterize developmental processes. So in this lecture, we're going to move on to developmental evolution. How do developmental processes change? In last lecture, we discussed how we can use the geometry, the topology of phase space to explain how a developmental process works, its dynamics. And today we're going to look at how it changes in evolution. And for that, I'm going to take a little sort of detour right at the beginning of this lecture, and we're going to introduce a very important concept here. And this is the idea of the Genotype-Phenotype Map. Okay, so this is a sort of a very abstract idea. It has its limitations, but it's very useful to sort of structure our thinking about the evolution of development. And what it does is it sort of focuses evo-devo on this problem of describing how you get from variation at the genotypic level to variation at the phenotypic level. So the basic idea is that you have an abstract space of different possible genotypes down here and an abstract space of possible phenotypes. We'll look in a bit more detail in a later lecture at how we define these exactly. But at the moment, all you need to know is that there may be specific sort of genotypes and they map to some phenotypes here, okay? So the arrows here represent the idea of a mapping. And this is hugely oversimplified. First of all, the first problem is that if you look at the evolution of life cycles, what you should really have is a phenotype-phenotype map. So you map from phenotypes to phenotypes in successive generations. That's what's actually happening. And the genotype is just sort of an important ingredient in that generational mapping. Okay, but still, it's an interesting idea. The other oversimplification is that, of course, there is nothing deterministic about this. There are a lot of sort of external influences on these arrows here that we're just sort of going to background and ignore for the moment. So you could have organismic sort of triggers, inductive signaling. You could have external environmental triggers that are important and essential for development to occur. Keep that in the back of your mind. And another thing that you should sort of think about when you think about this map, it's not a simple one-to-one mapping. In fact, we have sort of a two-way degeneracy, right? So there are arrows that go from the same genotype to different phenotype in different environments, for example. And this is the phenomenon of developmental plasticity. Depending on the environment, the same genotype can produce different phenotypes. The other way around also occurs. You can have different genotypes producing the same phenotype. We talked about this a lot when we talked about the correspondence problem, that there is not a single mechanism responsible and solely responsible for a specific phenomenon in development. And that, of course, is the phenomenon of robustness, or as Waddington called it, canalization of development. Okay, so the central sort of aspect of evo-devo, remember, if it is to be a mechanistic science like Günter Wagner writes, is to look at how variation is generated through development and how changes in developmental processes affect that variation that is available to selection. So the Genotype-Phenotype Map determines what we could call the variational property, and through them, the evolvability, the potential to evolve of a developmental system and an organism. So here's a quote from an absolutely famous and fabulous paper by Günter Wagner and Lee Altenberg in 1996, one of the big classics in the field. And it says, the variational properties of the phenotype are fundamental to its evolution by natural selection. Adaptation requires that genetic change be able to produce adaptive phenotypic changes in the first place. If you don't have phenotypes to select for, you don't have evolution by selection. Whether or not adaptive changes can be produced depends critically on the Genotype-Phenotype Map. So understanding the Genotype-Phenotype Map is crucial for understanding evolvability, and evolvability, as it has been claimed, by several authors is one of the central problems, if not the central problem of Evo, evolutionary developmental biology. So to get back to this idea of the genotype-phenotype map, we need an explanation of this map. And if you look at it, a mapping like this is sort of a mathematical concept. And if you're a mathematician, you can interpret it in different ways. So one way of interpreting a mapping like this is as a correlation between genotype space and phenotype space. And that is indeed what disciplines just like quantitative genetics do, and also genome-wide association studies. They are based on this interpretation of the genotype-phenotype map, that there is a statistical correlation between variation at the genotypic level and the phenotypic level, but it kind of black boxes what's going on here in the middle. And it should be abundantly clear by now in this lecture that this is not the sort of explanation that I want. Remember the XKCD comic about correlation and causation. What we want are mechanistic explanations of this map, okay? And these are causal explanations. So what we want here is a causal explanation of what's happening. And that's the sort of field of physiology and systems biology that's trying to fill the gap between those two levels of variation. Okay, so that is not just a central topic in evolutionary developmental biology, but also one of the central topics in evolutionary systems biology. So what is a causal interpretation of this map? Of course, we've already been through this. Waddington's diachronic biology tells us that this map represents generative processes, developmental trajectories, trios in Waddington's term, and generative process in the vocabulary of Brian Goodwin, who we will extensively discuss in the next lecture. So Goodwin says the aim here is to find out the generative principles, the organization of matter together with principles of directed or asymmetric time-dependent transformation. So we need to know the spatiotemporal arrangement of the system and the rules that guide its transition from state to state. And that's what dynamical systems theory does. We've been through this. Remember, last time I quickly sort of introduced the ideas of dynamical systems theory using this very simple example of the genetic toggle switch between the gap genes Krupel in green and Giant in blue. And I've introduced the abstract idea of a phase or state space that contains features such as attractors and depicts the flow of the system here, indicated by these arrows. And you can see that there are two basins of attraction and there are trajectories in these basins. The flow within these basins ends up necessarily at one of the two attractor points. And this explains why this system acts like a toggle switch. Either the green protein Krupel is present in a cell or the blue protein Giant, but never both, as you can see here in the middle of the embryo. Okay, so positive double negative is a positive feedback loop. It acts like a switch. And the switch-like behavior of this gene circuit is explained by the geometry, also called the topology of the phase space. So the sort of arrangement of these two basins of attraction, their attractors and the separators, the line between them. If we understand that sort of structure, the topology of phase space, then we understand what the system can do. Okay, and I've also introduced this idea that you can depict, for at least for simple systems with only two variables, you can depict this phase space as a landscape. We'll come back to that in a second. But first, also remember that there can be surprises. We cannot assume that the system reaches the attractor. So it only behaves in the predictable way that I've just said when it reaches steady state, the fixed points. If it's somewhere on the way there, it can have all kinds of surprising behaviors. For example, you can see here, and we've been over this in the last lecture already, that for quite some time, you can stay here. If you follow the trajectory, if you start with very close concentrations, you can see that both of the factor X and Y can coexist for quite a while, and only later on will the system start to act like a switch. So it will need time to do what it's supposed to do. And if you introduce a time delay in the interaction between those two factors, you can get crazy stuff happening, like transient oscillations in a model of a positive feedback loop. So negative feedback loops are usually associated with oscillations. This circuit should not produce oscillations, but under certain circumstances, it will. Again, the mantra of the lecture: network structure does not determine function. Network structure does not determine function. All kind of things can happen given a certain network structure. Okay, so this is all a repetition. And what we want to do is we want to take this depiction of the phase portrait as a potential landscape. Remember that as sort of an epigenetic landscape, which makes this metaphor, Waddington's metaphor, concrete. So this is a measurement of the topography of the landscape from observables. It's no longer a metaphor. It's something we have measured and extracted from the data, even though we are using modeling here. It is an empirical approach to identify the attractors, the basins of attraction, the systems. And I was arguing that these features of phase space are as real as anything else that we're using. So they really exist. They govern the behavior of the system. And so they're robustly detectable, describable, derivable in the sense of WIMSAT's sort of trustworthiness, their trustworthy knowledge about the real world. Okay, I also quickly introduced last time Waddington's second most famous diagram, which is the underside of the landscape, represents the genes, as pegs, and then this complicated network of guy ropes that connect to the landscape. And the idea that Waddington had, that if you change the genes by mutation, random mutation, you will get some really complicated effect on the landscape. And what you need to do, if you read the strategy of the genes, the central aim of evolutionary developmental biology is to understand how this landscape changes over time. So we need to understand, if we tune the system, if we change, for example, the interactions between genes in the system, how does this landscape change if we want to understand developmental evolution? This is what we're going to do now. So just very briefly to remind you of the formalism, here is a drawing of this toggle switch model. Remember that not only are there two repressive interactions between X and Y, but we've also had, we allowed for auto-activation of these two factors and some inputs, activating inputs from outside the system, which are called the boundary conditions of the system. And you have all kinds of parameters that describe the strength of the interactions, and also the half-lives, the decay rates or the half-lives of the protein, so how stable the proteins are. These are parameters of the systems, while the concentration of both of these factors are the state variables. And I had said, if you do a simulation, usually the parameters and boundary conditions, which are also some sort of parameter, stay the same, they stay constant, while the variables, as their name says, they change over time. So what happens, we can ask now, if evolution works on this circuit? And for example, what happens if evolution changes the strength of these interactions? So what we're going to do now is we're going to vary the parameters and see what happens when we do that. And the toggle switch model is a perfect toy model to think about what is possible in evolution. So how can this toggle switch model evolve? How can evolution affect it? So if you have random changes in the parameters of the system, what can happen? And there are exactly 4 different possibilities. The first is evolution changes somehow the initial conditions of the system. So imagine that usually you start with more green protein in a specific nucleus than blue. You always end up at the cruple on, giant off attractor. Okay, but it could be that now evolution changes the concentration. So cruple and giant concentration increases by a lot, but still there's more cruple than giant. The system will still converge to the same attractor. And I was telling you that this is a way to explain the canalization, the robustness, the resilience of development towards perturbation, genetic and environmental perturbation. Okay, another possibility though is that suddenly you have a bit more of the blue protein than the green protein, and then the the state of the system will suddenly flip. It will end up at high giant, little Krupel instead of the other way around. The switch has switched, okay? So there's one possibility here. The second possibility is happens, for example, if you change the strength of the boundary conditions, the activating input. So if you make that activating input weaker, you will end up with less of each protein, less giant, less Krupel. So the weaker these interactions are, the further together, closer to the origin down here, these attractors come to life. Okay, so they sort of converge towards the origin here, the further you tune those activating inputs down. So here, what's happening here is you have a change in attractor position, but the sort of qualitative nature of the dynamical regime here is still the same. It's still a switch, basically. The behavior is qualitatively the same. The third possibility is, for example, if you make repression asymmetric, one of the repressive interactions becomes much stronger than the other. What you do is you shift the separatrix. And actually, it now looks like this. So if you start at the same point, you suddenly converge, even though you start with more Krupel than giant. If giant is repressing Krupel more strongly than the other way around, you will end up at the other, the giant on, Krupel off, steady state, the attractor. Okay, so you can change the system by changing positions of separatrices between basins of attraction. And so this still is the same sort of relative arrangement. It still acts as a toggle switch, but now you have a bias towards ending up with high giant versus high Krupel. Okay, so the last and most interesting possibility happens, for example, if you tune up the autoactivation strength. So if you make the autoactivation of both of these factors stronger, we'll do it symmetrically, so both of these autoactivation terms get stronger, and what happens is something very surprising. Suddenly, you get a new attractor that comes up here in the middle. Actually, what happened is that the saddle point that was here in the middle turned into a stable attractor and spawned 2 additional saddle points. This is called a pitchfork bifurcation. It's a bifurcation because there is a new attractor that is created. Sometimes bifurcation annihilate attractors too. I couldn't put that in because of space in this slide. Okay, so the creation or annihilation of attractors is called bifurcation. There are different types of bifurcation. In this specific case, it's called a pitchfork because it starts from one saddle point, sort of spawns 2 saddle points, and in the middle creates a new stable state. So if you draw it in parameter space, it looks like a pitchfork. Okay, and so this is what we'll do next. We'll look at how the system behaves. We're trying to systematically look at how the system behaves, this toggle switch model, when we tune the parameters randomly. But first, a few more thoughts on this. So we have 4 different ways in which random changes in the parameters, including the boundary conditions, can affect the system. Okay? And those 4 are very interesting because if you think about it, only this one possibility here leads to gradual change. Okay, so only in this case do you get gradual phenotypic change. All the other ones are so-called threshold effects, where you get a sudden switch from, because this is a switch-like system, you switch from high Krupel to high giant without intermediate state. So this is a discontinuous threshold-like phenomenon. So if you only have very small gradual changes in the genes, in the genotype of the system, that is no guarantee that you will get a gradual smooth change in the phenotype of the system. And in fact, this theory from first principle tells you that you have to expect discontinuous changes in phenotypes in evolution. Evolution cannot produce all the changes that we see in nature without such effects, okay? Based on first principles. In particular, we're going to focus now on this idea of evolution through bifurcation because it's the best worked out part of the theory. And so what you can do is you can systematically draw the strength here of the self-activation. We're going to look at this bifurcation in a bit more detail in a more systematic way. So what we can do is we can draw the strength of the activation, and these lines represent the different attractors in the system. So what is happening here is, as we have very low activation, we tune it up. We have two attractors, one with low X is giant, right? Low giant, high giant, and then suddenly you get this new attractor state here. So you get this pitchfork bifurcation that we were looking at before, and you get a new attractor state that has intermediate concentrations of both factors coexisting, and a gray basin of attraction that pops up as well. So the saddle has been transformed into a new attractor through a pitchfork bifurcation. Now, what I haven't told you before is that if you tune the auto-activation up even further, it becomes dominating in the system, okay? And so the switch-like behavior goes away. Again, structure does not determine function. So you can have this switch, toggle switch motive, but if those auto-activating terms around it are strong enough, the system cannot switch anymore. So what's happening here is we have two bifurcations at the same time. They're called saddle node bifurcations. You don't have to worry about what that is. Also called blue sky bifurcations, because they create attractors out of nothing, basically. Okay, so what you have there is the sudden disappearance of those two purple high, giant high states, and the only state that remains is the one where both factors are present at low concentration. Okay, so we have systematically looked at what happens if we crank up autoactivation, and we find three different regimes, dynamical regimes here. One, bistable, that's the typical switch behavior. That's what we expect. In between here, a tristable state where you have an additional attractor in the middle, and then a yellow monostable region where you only have this attractor, and both factors persist at equal concentrations. So the switch no longer switches in this regime. Okay, and so if we look at this systematically, we can see that you can get from all of those regimes to any other regime, okay? So here's from the bistable to the tristable regime is our pitchfork bifurcation that we've already talked about. And then from the tristable to the monostable regime, you can get saddle node bifurcations, but you can also get a direct sort of jump from a monostable to a bistable regime. Again, that's a pitchforks bifurcation. So you can turn all of those regimes into each other by tuning the parameters of the system. So if you get random changes in those parameters, you will go through all those dynamical regimes eventually. Okay, and so this is a toy model, so it's very simple, but people have looked at models of more realistic system. For example, here, a very early example of wonderful paper from 1982 by George Oster and Pera Albert that looked at realistic sort of evidence for such behavior. And they found that there is a model of slime mold, Dictyostelium aggregation, that was doing this sort of thing. Okay, so this is from a paper by Goldbeater and Sagel from 1980, a classical model of slime mold aggregation. And you don't need to understand the details, but basically what you see here is a sort of a two-dimensional version of that one-dimensional bifurcation diagram I was showing you before. And the letters A, B, C, D, and E, they indicate different dynamical regimes of a biochemical model of CAMP secretion in this system. So you can have different kinds of behaviors, and these lines here are where bifurcations occur. And so these lines are called the bifurcation set. It's a set of points. They're called the bifurcation set of the system. So basically the idea of Oster and Albert's theory of evolution, which we'll get into in the next lecture in more detail, is that if you can reconstruct those bifurcation boundaries, then you know what kind of transitions can occur and how likely they are to occur. So observe here that, for example, you cannot get from A to D directly. So you have to go through B and C or through E to get from A to D. So the transition from A to D is impossible in principle, not possible, okay? And also you will see that, for example, to get from E to B, there's a very small bifurcation set here, a very small edge. And that means it is much less likely to happen if you change the parameters, the genes randomly. This is much likely to happen than, let's say, a transition from A to B or from E to D, which have much larger bifurcation sets. So basically, this is a theory that can give you the probability of phenotypic transitions from random genetic changes. And you could think of sort of a cartoon version again of a parameter space here, a bifurcation diagram. You have different dynamical regimes and two species populations living, one here in A in this region and one here in D. And you can now, and this is a revolutionary proposition by Pera Alberg in a wonderful paper from 1991, is you can draw a sort of a transformational diagram. You can draw which one of those dynamical regimes can transition into another one and which ones cannot. So you can't get directly from A to C, for example. There's no arrow here, right? And then you can map those species onto that diagram, of course, and you can say, look, if you're species A, the only thing you can do is you can become B or D, but you cannot directly go to E, C, or F, okay? And so on and so forth. So here, A can only go to B or D, and D has much more potential for transition because it's in this central area here, D, so it can transition into all the other regimes. So it has a larger evolutionary potential. It is more evolvable than species A. So if you could work this sort of structure, these bifurcation diagrams out for real systems, you could have a predictive theory of how likely a phenotypic transition is. And that gives you what Alberg calls a map of the possible for evolution. It's basically the substrate on which phenotypic variation is produced and on which selection can act. So to bring together last lecture and this lecture, basically, what I was saying is that you have a sort of a time scale of development here. You have state variables like protein concentrations, etc., and systems parameters that, you know, interaction strengths or the nature of interactions, decay rates, etc., diffusion rates, whatever you want. And so only the state variables change while the system parameters stay the same at the level of development. Okay, at the level of evolution, at the larger time scale of evolution, you have the parameters become the state variables. So you change the strength of the interactions, you change the decay rates, and the system parameters now become things like rates of evolution, okay? The reliability, fidelity of inheritance, the persistence of heritable signals, etc., etc., etc. And these two time scales interact. Development produces phenotypic variability for selection to work on, and of course that alters the selective pressures on the system. So this is a very sort of simple version, and it's a little bit too simple, okay? Because remember, the sort of, well, you know, this is only one, this sort of bifurcation view is only one way in which evolution can change the system. And we'll come to a further complication, that of transient behavior, in a minute. But let's see what you can do with this. So let me give you a classical example of how this was applied by Pera Alberg in his experimental work, okay? So what Alberg looked at together with Gale, so they looked at the development and evolution of limbs, of digits, okay? And in particular, they studied salamanders and compared them to other creatures. And they found something really interesting, okay? So salamanders have, just like, for example, hoofed mammals, have a reduced number, often a reduced number of digits. And so if you compare different salamander species, you'll find a stereotypical pattern of digit reduction. So a normal let's call it a normal salamander hand here in this species has 4 digits. You can number them 1, 2, 3, 4. And then as you compare to other species, the first digit that is reduced is number 4, and 3, and 2, and the last one remaining is the first, which would be the thumb in our case. While if you look at hoofed mammals, so you have a pig here, a sheep, the digit reduction happens in a different order, okay? The first one to go is number 1, all right? And then the next one to go is number 5. And then number 2, number 4. And the last one to go is the middle sort of finger. I'm not going to show it to you right now. But this one is in unpaired hoofed mammals is the 1 digit that remains. So there's 2 very different sort of sequences of threshold-like reductions, okay? So each digit disappearing is like a threshold happening in the limb butt of the animal, okay? So this is one of these threshold effects, and Albert is sort of is imagining, is speculating that there is a bifurcation that's happening in the limb field upon each one of those reductions. What's really interesting is that if you perturb those digits in a species experimentally, you'll see exactly the same sequence. So if you perturb development of salamanders, okay, so what goes first is the 5th or the 4th digit and so on. And the last one remaining is number 1. While if you do this in pigs or sheep or frogs that also have this other pattern, so the only animal group that does this are salamanders that have this unusual pattern, okay? So it's 1, 5, 2, etc. Okay, a completely different pattern of reduction. So basically, the developmental sequence is the inverse of the loss pattern in phylogeny. Okay, so the sequence of loss in phylogeny in experimental situation is a classical threshold effect, just like I was introducing before, is determined by the structure, not the network structure, but the set of rules. Remember, we said the structure of a process is the set of rules that generates it. So what's happening here is not determined by selection. Which digit disappears is not an adaptive feature, but it's determined by the set of rules by which these animals grow their digits. And the difference is that you have a different sequence of bifurcations during development that produce these different digits in salamanders and other vertebrates, okay? So this is revolutionary work in the 1980s. First the experimental work, and then the sort of dynamical systems interpretation by Pera Alberg in collaboration with George Oster is sort of a revolutionary, it's called a structuralist interpretation to set it apart from an adaptationist sort of perspective. And again, structure here doesn't mean network structure, but it means the structure of the process, the developmental process, the set of rules of the generative process that underlies the trait. Okay, so this is how you can use bifurcation theory to explain evolutionary phenomena. Classical example, fantastic example, but the situation, as usual, is more complicated. Remember that bifurcation was only one of 4 different ways in which you can affect a dynamical system in evolution. And also, remember from the very beginning of this lecture that I told you that often we cannot assume that these systems are at the steady state. So they're somewhere in transience to that state. So transient behavior, the behavior of the system before it reaches a steady state, can be very important. So what my own research group has done a few years back is sort of think about this. The reason that people always look at steady states and bifurcations is you can apply established mathematical theory to that. So if you remember back to my lecture about how we use models as tools, I was telling you that these models are always constrained by the frameworks that we use to formalize them. This is Tarja Knutila's work. And so here we are constrained to look at situations where we are at the steady state, at the attractor of the system, because we only have the mathematical tools to look at those situations. So what we've done in my research group a few years back is look at transient patterns in time variable systems. So systems where the parameters change. Okay, so you could imagine that this is an evolving developmental system, or sort of a system that changes during developmental time, maybe through the outside sort of effect of an inductive signal or an environmental trigger. Stability of proteins changes, the interactions between the factors change. And what happens here, it becomes very difficult to do this mathematically. So we did something very crude. We just simulated all these sort of different scenarios in this toy model of the toggle switch, and we classified them according to the different sort of geometries of the trajectories that we saw. So what you do is you simulate the system and you draw a different potential landscape for different points in time. So what's happening now is the landscape becomes a seascape. It starts moving around and changing as you simulate the system. And what we're doing is we're stopping the system at given points in time, and we assume we do the analysis as if this was the system, it wouldn't change. And so then we get a sort of, instead of one portrait of the face portrait, we get a whole sequence of face portraits that change over time. So this may be a little bit confusing. Don't worry about the mathematical details. What it looks like is as follows. So the classical situation is the system is at a steady state here, at this one in particular, and then you suddenly get a bifurcation because you've changed something, like you've added autoactivation. And from that point on, you know, there's a different, the different set of bifurcations here. So this steady state at which the system was destroyed, and it now transits to a different steady state. Okay, so this is a classical situation. A bifurcation occurs. The system is at the steady state, and it changes from one attractor to the other. Okay, this is what I just described, the whole last 20 minutes. But there are different possibilities. Here is one where the system is still rolling down the landscape, and then the bifurcation occurs when the system is still very far from the steady state. And now it sort of gets caught. There's a capture in a new basin of attraction. And so we called this a topological capture because the topology of the phase space had changed because of a bifurcation. Remember, bifurcations change the relative arrangement, the relative number of attractors, and so they change the topology of the landscape. Okay, so you can come up with a bunch of different behaviors, and we classified them into 4 types of behavior, which gives us a tool for understanding the different types of dynamics you can get from this system. Remember, way back when we talked about process thinking, I said it is possible, just like you can distinguish objects and put them in different classes. This is an apple. This is an orange. They're not comparable. Okay, you can also classify different processes into different categories. And so we can do this based on these different behaviors. Here are the 2 that I talked about already: transition, topological capture. You can have something called a geometric capture, where the separatrix is moving and captures the trajectory. You can get loopy sort of trajectories here that you cannot get in normal dynamical systems. And you can have a system chasing a moving attractor here. We call this a pursuit. But basically, all of these different types of dynamic transient time-variant behaviors generate different types of dynamics, okay? And different types of transitions that you can get from those dynamics. So basically, what we've done is we've built up a huge toolkit to look at the dynamics and the evolution of developmental processes using dynamical systems models. This is all I need you to remember. We have a great toolkit to look at these processes, to describe them in terms of their attractors, their bifurcations, and their transient behaviors. We can classify them into different groups, and we can now check if we can explain some of the phenotypic transitions between forms or patterns in nature using this theory. So the whole idea here is captured in this beautiful movie that I will end on, and that is Waddington's landscape is not fixed. You see, as these sort of variable balls here roll down the landscape, they are making the valleys as they go along. So there is no separation. I said that from the beginning, between the ball and the landscape. They're one system that feeds back. The ball and the landscape feed back on each other. okay? And so as these balls roll down the landscape, they create their valleys in which they end up being. So it's a seascape, it's malleable, and the ball and the landscape are tuned to each other. And we have the mathematical tools. We have a cartload of tools now. We have all the ingredients we need to build a very, very complicated theory of organismic form. So the next lecture will look at how do you apply these tools that we developed in the last 3 lectures to a theory of biological form and phenotypic transitions. And we will trace a history of attempts that have done that. Okay, so I hope you tune in again then, and wish you a nice time. Until then, goodbye now.